A'udhu billahi minash shaitanir rajim. Bismillahir rahmanir rahim. Hello, dear ladies and gentlemen. In today's lecture, we will discuss about genetics and the basics of genetics. So, first of all, we are coming to discuss basics of genetics. Etiology of a disease in general may be environmental, genetic, or both of them. Fully genetic disease are common. 50% of spontaneous abortion during early gestation have demonstrable genetic abnormalities. Now I am coming to discuss with you terminology in genetics. Hereditary disease derived from parents, familial disease transmitted from one generation to the other through generation can appear in any one of the following. Congenital disease, parent at birth born with not always genetically determined, e.g. congenital spills, toxoplasmosis. Not all genetic diseases are congenital, e.g. Huntington disease appear in third to fourth decade of life. Now I am coming to discuss with you genotype, the genetic composition of a specific character. Phenotypes, the observed function and morphological characteristic caused by genotypes. Now I am coming to discuss with you alleles. So what is allele? Allele is one part of a pair of a gene that appear at a particular location on a particular chromosome and control the same characteristics such as blood group or color blindness. Now I am coming to discuss with you gene. So what is gene? Gene is the part of a DNA sequence that code for a specific character and is composed of two alleles. Chromosome and genetic information. Genetic information is stored in the DNA sequence and found on the chromosome. The typical normal human cell contain 46 chromosome, i.e. 23 pair of chromosome, 22 homologous pair of autosome, and 1 pair of 6 chromosome. 6 chromosome may be XX female or XY male. Member of a pair of a chromosome described as homologous chromosome carries matching genetic information, i.e. they have the same gene Lucy in the same sequence of nucleotides. Now I am coming to discuss with you gene through at a specific locus that may have either identical or slightly different form, which are called alleles. So what is called allele? Allele are the part of a gene which are present on a specific part of a gene and show one similar characteristic. One member of each pair of a chromosome is inherited from the father, the other from the mother. This is a diagram of the chromosome. The first two are short arm, the central part are centromere, and the two long arm are chromatid. Now I am coming to discuss with you DNA structure. So what is DNA structure? Each molecule contains a continuous DNA deoxyribonucleic acid molecule. DNA is composed of two very long complementary chains of a deoxynucleotides. The two chains of DNA wind around each other, i.e. twist about each other, forming a double helix. The twisted ladder model. Each deoxyribonucleotide in turn is composed of a nitrogenous base, i.e. adenine A or guanine G or cytosine or thymine bound to deoxyribose and phosphate. DNA function, it codes for the protein which are important for the structure and function of the cell. It provides the genetic information for protein synthesis. It transmits the genetic information to the daughter cell and to the offspring of the individual by a process of transcription and translation. So now I am coming to discuss with you genetic disorder. So what is genetic disorder? Can be categorized into monogenic, Mendelian disorder of one gene defect, mutation affecting one gene result in a new change in function or morphology. Now I am coming to discuss with you multifactorial disorder. So what is multifactorial disorder and what are the causes of it? Due to polygenic factor, maybe multiple gene or more than one environmental factor. Chromosomal aberration, abnormal in number or structure of chromosome. Mitochondrial disorder, this type is mediated by maternally transmitted mitochondrial gene which are inherited exclusively from the maternal side. Sperm has no mitochondria unlike ova. It is a rare form of inheritance mentioned here just for the sake of completeness. Now I am coming to discuss with you monogenic disorder controlled by one gene. Today there are 50,000 diseases inherited as a Mendelian pattern which can be divided into autosomal dominant, Autosomal recessive X-linked disorder, one autosomal dominant disorder. Only one allele is needed for the character to appear. Now I am coming to discuss with you dominant character. So what is mean by dominant character? Dominant character mean only one allele is enough for an individual to be affected. So both homozygote and heterozygote are affected, i.e. no carrier state. Every affected child should have at least an affected one parents. Some patients do not have affected parents because of new mutation in the sperm ovum from which the parents are derived. Mutation occur more frequently with parents of advanced age. In the mating of an affected heterozygote to a normal homozygote, the usual situation, each child has a 50% chance to inherit the abnormal allele and be affected, and a 50% chance inherit the normal allele with no disease. The two sexes are affected in equal chances because the defective gene resides on one of the 22 autosome, i.e. non-sex chromosome. Expression of autosomal dominant gene. Autosomal dominant disorder commonly shows variable expressivity. Variable expressivity is the ability of the same genetic mutation to cause a wide phenotypic spectrum. It is when the traits is seen in all individuals carrying the mutant gene, but it is expressed differently among them. For example, some parents with neurofibromatosis type 1, which is an autosomal dominant disorder, have only brownish spots, calf outlet spots, whereas other patients with the same genetic constitution have a severe form of the skin tumor and skeletal deformities. Therefore, neurofibromatosis is said to show variable expressivity is affected individual. Now, I am coming to discuss with you example of autosomal dominant disorder. Marfan syndrome, Ehlers-Danlos syndrome, osteogenesis imperfecta, achondroplasia, Huntington disease, neurofibromatosis, tuberous sclerosis, myotonic dystrophy, familial hypercholesterolemia, hereditary spherocytosis, familial polyposis coli, polycystic kidney disease. Now I am coming to discuss with you autosomal recessive. In autosomal recessive disorder, the phenotype is usually observed only the homozygote 2 allele. The typical pedigree shows affected male and female sibling with normal parents. Parents are carrier. Recessive inheritance is suspected when parents are consanguineous in the mating of two character heterozygotes who are phenotypically normal. Who are phenotypically normal. The segregation frequency with each pregnancy is 25% normal, homozygous 25% affected homozygous, and 50% carrier heterozygous, which are clinically normal. Children who have an autosomal recessive disease frequently show signs and symptoms early in life, whereas many autosomal dominant disorders have delayed onset, e.g. Huntington disease clinically manifests for the first time during adulthood. Example of autosomal recessive disorders are sickle cell anemia, thalassemia, congenital adrenal hyperplasia, cystic fibrosis, Wilson disease, hemochromatosis, enzyme defect disorder, phenylketonuria, galactosemia, homocystinuria, lysosomal storage disease, alpha 1 trypsin deficiency, glycogen storage disease. Now I am coming to discuss with you sex-linked disorder. All sex-linked characters are carried in the female and appear in their son. This is because the Y chromosome is short and carrier no character. and it is only responsible for the determination of male gonad sex factor. So the presence of one allele in X chromosome is enough for the phenotype to appear in male. That this type of inheritance is suspected. So, dear ladies and gentlemen, our time is finished. So, in next lecture, we will discuss from female disorder of due to genetics. Goodbye, dear ladies and gentlemen. If you like, please subscribe and share with